What I'm going to talk about is this enzyme called methylthioadenosine phosphorylase. Um, it's kind of a important enzyme because the substrate methylthioadenosine is generated from polyamine biosynthesis. And it turns out that most of the adenine in cells, which is used for RNA and DNA synthesis, ATP synthesis, comes from salvage, which is due to this particular enzyme, which generates adenine as well as methylthiouriboside 1-phosphate, and which is then converted in a series of steps to methionine. So cells that lack MTAP turn out to be methionine oxytropes, and also have defects, not defects, but now have to rely on de novo purine synthesis to generate nucleotides. So this bottom slide shows the lesion which occurs with MTAP deficiency, and that is now there's an increase in this coenzyme PRPP, and this allows bases like fluorouracil and 6-thioguanine to be converted even more efficiently to nucleotides and be more potent anti-cancer drugs. Where is MTAP located? What's important about this is that it's frequently co-deleted with the INK4A and INK4B at the 9p21 locus, so that this is a very common deletion in cancer, and MTAP is often co-deleted and makes it a little difficult to distinguish between the effects of MTAP deletion and these tumor suppressors. What's important about MTAP is that it's deleted in a large number of cancers, and there are a couple of cancers in particular have a very high percentage of MTAP deficiency. Mesothelioma in particular, pancreatic cancer. There's new information in glioblastomas. It's deleted almost in 50% of these tumors. Pancreas have a high level of MTAP deficiency. And in Tcell leukemia, lymphoma, there's also a very high level of MTAP deficiency, and that's an area in which we're particularly interested in. Now, the question is MTAP a tumor suppressor? And there's some evolving evidence that it may be, and that the effects of this p16 deletion, homozygous deletion, is not only due to the INK4A and B deletion, but also MTAP deletion. And some of this evidence is that there's instances where MTAP deficiency is specifically deleted. And there's also evidence that MTAP can be downregulated by promoter methylation. Secondly, Warren Kruger showed that in knockout mice, homozygous mice, it was embryonic lethal, but heterozygous MTAP knockout mice developed Tcell lymphomas late in their lifespan. Finally, transfection of MTAP into MCF7 breast cancer cells, which lack MTAP, resulted in the inability of the cells to form tumors. So there's evolving evidence that this is a tumor suppressor in itself. And there's also data that shows that homozygous deletion of MTAP are more frequent than p16 for a homozygous deletion in non-small cell lung cancer. And a recent paper shows that it's an independent prognosis marker, and the concordant loss of MTAP and p16 expression predicts short survival in non-small cell lung cancer. So clearly it has an important function as a tumor suppressor. In lymphoma, in non-Hodgkin's lymphoma, the INK4A locus is deleted in a high percentage of non-Hodgkin's lymphoma patients. We don't know about MTAP specifically, but presumably MTAP is co-deleted in a high percentage of these patients, and also especially in the transformed aggressive variants. In Mantle cell, there's an interesting study in which they looked at MTAP expression in Mantle cell tumors, and although a small percentage of these patients had deletion of MTAP, turned out when they looked at the survival of those patients, it was markedly decreased as compared to the patients who had whose tumors had expression of MTAP. Even back in 1981, Denis Carson published a paper in PNAS showing that MTAP-deficient cells are more sensitive to de novo purine synthesis and methionine deficiency. And that observation has been reproduced many times in different laboratories, including our own, and yet there was only one clinical study in which patients with solid tumors were treated with L-alanisine, which is an inhibitor of IMP to AMP biosynthesis, in patients who had MTAP specifically deficient cancer. And of about, I think there were 40 patients here, there were no objective responses. 24% had stable disease, 2 patients with mesothelioma. The conclusion was at this dose and schedule, L-alanisine was ineffective in patients with advanced MTAP-deficient tumors. So why didn't it work? Well, they only treated solid tumors. And L-alanisine is an ineffective drug, and it's been abandoned. Phase 2 trials didn't show any activity. So they used an inactive drug in the usual dose schedule, and they didn't see anything. They didn't do any pharmacodynamic studies to show that the drug was actually inhibiting AMP synthesis in the tumor. Did it really get into the tumor? And finally, there may be that there's some salvage of adenine or adenosine by the stroma, and which prevented its activity. So as I said, we're interested in T-cell lymphoma. Recently, we published a little paper showing that even the uncommon lymphomas have a high incidence of MTAP deficiency. Angioblastic lymphomas, anaplastic large cell, and as you can see, they're very high incidence of MTAP deficiency in lymphoma, so that we have pursued studies in T-cell lymphoma in our laboratory. This is an example showing that if we take a cell line which lacks MTAP, the CEM cell line, and compare it to another cell line which has MTAP, there's a marked difference in sensitivity to 6-thioguanine, a de novo inhibitor of purine biosynthesis. There's also somewhat of a difference in this compound pralatrexate, which is a methotrexate analog, which is used to treat T-cell lymphoma. And so we've used pralatrexate in our studies. And the reason for that is that, as Bruce Chapner showed many years ago and his colleagues, that methotrexate and presumably pralatrexate are converted to polyglutamates in cells, and they then become inhibitors of de novo purine synthesis. We've also knocked down MTAP in the MOP4 cells, and they become sensitive, and we've added MTAP to CEM cells, and they become more resistant. So what about in vivo? Phil Tedeschi, our graduate student who's here, looked at the effect of pralatrexate and 6-thioguanine in tumors, in xenografts in the mouse. And as shown in this first part of this slide, the CEM cell line is much more sensitive to pralatrexate than is the MOP4 cell line, the one that has MTAP. And surprisingly, the MOP4 cell line actually grows in the presence of 6-thioguanine as compared to CEM cell line, which is still very sensitive to 6-thioguanine in vivo. So we thought, well, maybe we can use a combination of 6-thioguanine and also pralatrexate in cells. And so when we used optimum doses of these drugs, we got very nice tumor regression. But as you can see in the green line there, the tumors really disappeared rapidly, but the animals died because it was a toxic combination. So by looking at different dose schedules of 6-thioguanine and adding leucovorin rescue to the pralatrexate, we would then develop a safe and effective schedule in the CEM xenograft cell line. And I won't go through this with you, but basically by adding leucovorin and decreasing the schedule for 6-thioguanine, we could get great tumor regression without much toxicity. Our conclusions are that MTAP tumors are more sensitive to pralatrexate and 6-thioguanine than MTAP positive tumors. The combination resulted in improved tumor response, and that was due to the fact that we used leucovorin rescue. In another study that we published recently, we showed that leucovorin rescue, like rescue of methotrexate, leucovorin rescue for pralatrexate is even more effective, causing decreased toxicity without compromising the antitumor effects. And a shorter course of 6-thioguanine allowed the combination to be better tolerated. And so we have now generated a clinical study using this combination, which has just been approved by IRB. The clinical study, the rationale is what I've already enunciated, that pralatrexate, when polyglutaminated, inhibits de novo purine synthesis. Leucovorin rescue decreases pralatrexate toxicity, allows higher doses of pralatrexate to be administered, and 6-thioguanine inhibits de novo purine synthesis. In the clinical study, we'll use a fixed dose of pralatrexate, and starting with low doses of 6-thioguanine, we will increase the 6-thioguanine dose to get a MTD, and it'll be in patients with refractory Tcell lymphoma. Part 2 of this presentation is that can we protect normal tissues, but not MTAP-deficient tumors, from 6-thioguanine with methylthioadenosine pretreatment to allow higher and more effective doses of 6-thioguanine and pralatrexate to be administered? And this idea was generated by our colleague Martin Lubin, who's in the audience today. So again, the same slide shows the rationale for this, and that is that in the presence of methylthioadenosine phosphorylase, if one gives methylthioadenosine, you load up the cells with an adenine. As a consequence, adenine, you have a higher concentration of adenine in cells. It uses up PRPP, so there's less available for the conversion of 6-thioguanine or fluorouracil into nucleotides. So MTA pretreatment then decreases the amount of nucleotides in cytotoxicity of these drugs. In the absence of MTA, there's less PRPP used by adenine, and in fact, fluorouracil and 6-thioguanine are more effectively converted to nucleotides. So does it work? This is a study that Martin Lubin and his son did several years ago. And it shows that by pre-treating mice with methylthioadenosine, 50 milligrams or 100 milligrams, one can protect mice from a lethal dose of 6-thioguanine. In the presence of MTA, 50 milligrams per kilogram, a lethal dose of 6-thioguanine is completely obviated. And even 2 doses of 6-thioguanine, days 1 and 4, if one precedes this by 100 milligrams per kilogram of methylthioadenosine, the animals are protected from lethal toxicity. So this small experiment shows that not only can you protect from a lethal dose, but by even can protect super lethal doses from 6-thioguanine toxicity. In another experiment done by our colleagues at Southern Research, a small animal toxicity experiment, showed that you can also protect from a lethal dose of fluorouracil by pre-treating the animals with methylthioadenosine, shown here, in either giving the drug before fluorouracil or giving the drug at 2 different schedules, before and after fluorouracil. So does this, can you show this actually in these 2 cell lines I described before, one which has MTAP and one lacks MTAP? And so this slide very nicely shows that the CEM cell line, which lacks MTAP, by giving methylthioadenosine before 6-thioguanine has no effect on the lethality of the 6-thioguanine. In contrast, the 4-cell line, which has intact MTAP, is protected from the lethal effects of 6-thioguanine by pretreatment with methylthioadenosine. So this is kind of a surrogate for normal cells. So in an experiment that we published in Cancer Biology and Therapeutics, we did the same experiment in mice bearing the CEM tumor. And what this slide shows is that mice treated with MTA alone or control mice, tumor grows very rapidly. And starting treatment when the tumors were fairly large with 6-thioguanine alone resulted in good tumor regression, but the animals died from toxicity, given every 4 days for 3 doses. By pre-treating the animals with methylthioadenosine, the animals were able to, we showed good tumor regression, but the animals survived very nicely from this lethal dose of 6-thioguanine. And if one looked at the toxicity, the combination of methylthioadenosine and 6-thioguanine in this case was essentially non-toxic as compared to the 6-thioguanine alone, which caused the animals to die from toxicity. In another experiment, we wanted to know whether we could use the 2 drugs together and get the same result. And in this experiment, animals were treated with the combination of 6-thioguanine plus or minus methylthioadenosine, shown here, or pralatrexate alone. And what this shows is that by giving 6-thioguanine 5 doses with pralatrexate in 2 doses, very much like our clinical study we're proposing, the combination is lethal without methylthioadenosine. With methylthioadenosine, we get very nice tumor regression. And this is labeled wrong, but if one looks at the weight of the animals treated with the 2 drugs together, or the 2 drugs pre-treated with methylthioadenosine, you can see that the animals tolerated the treatment very well without any marked weight loss. Another study which is quite interesting in terms of the use of this combination in solid tumors was published in 2012 by the group in Canada, Dr. Collins and his colleagues. And they were interested in sequencing prostate cancer cells with the next generation sequencing. And one of the patients of the 50 they sequenced turned out to have a methylthioadenosine phosphorylase deficiency. And they contacted Martin Lubin and myself because they saw our paper with the MTA protection of 6-thioguanine toxicity. And it turned out this patient's tumor was a neuroendocrine tumor. And what was interesting was that they then sequenced another 8 neuroendocrine tumors in prostate, and 5 of the 8 turned out to have MTAP deficiency, whereas only 1 of 40 or 50 non-endocrine prostate tumors had MTAP deficiency. So they were able to grow this tumor in the kidney capsule. So they, as I say, contacted Martin Lubin and myself, and they used the same dose schedule that we use with MTA protection in these kidney capsule tumors. And what they found was that just using this exactly same schedule, comparing it with saline control, they were able to get growth inhibition of these neuroendocrine tumors in the kidney capsule, and showed that there was an increase in apoptosis by caspase-3 expression in the tumors that were treated with MTA and 6-thioguanine. Now, of course, this is only one dose schedule. They didn't increase the dose of thioguanine, and so that by optimizing things, they possibly could have even done a little bit better. What about 6-thioguanine? Well, this comes from the publication in The Oncologist. And 6-thioguanine has been pretty much of a failure in patients with solid tumors. We looked at the literature, and almost all the studies were done with a single dose schedule, 5 days of 6-thioguanine given parenterally. And there were a few responses, but in general, as you can see, except maybe for breast cancer where there are a couple of complete remissions, 6-thioguanine in the dose and in the schedule they used was essentially inactive. So the question is, can you do better if you were able to increase the dose of 6-thioguanine effectively by using methylthioadenosine pretreatment? So our proposed follow-up protocol, if we can get somebody to make methylthioadenosine for us, is to use the same protocol using methylthioadenosine pretreatment followed by 6-thioguanine and pralatrexate leucovorin. And the question is, will the increase in doses that you can give, and we don't know how much of an increase in dose is going to be tolerated, will that enhance the tumor response? And the second question is, will the increase in doses be effective in solid tumors lacking MTAP? I think these are important questions, and that we hope to stimulate others as well as ourselves to try to answer these questions. So just to end up on this, and just to emphasize what I just said, what dose is best to use, what doses 6-DG can be tolerated using MTA pretreatment, are 6-thioguanine plus or minus pralatrexate the best de novo purine synthesis to be used. And what about this salvage issue? Would salvage from ATP and adenosine decrease effectiveness? Adenosine is not around very long in cells because it's has a very marked effect on cells. Would a nucleoside transport inhibitor increase the effectiveness? So thank you for listening. Phil Tedeschi, the graduate student, did most of this work with 2 Rutgers students, Yamini Kathari and Ikra Faroki. Nadine is our technician, and Martin Lubin has been a co-collaborator and co-investigator in these studies. And this is a picture of Phil and the 2 graduate students, Ikra and Yamini. This is the way we go to work every day at the Cancer Institute of New Jersey. Thanks very much.
So, Joe, it's really interesting. We've always been puzzled by the fact that purine antagonists are so active in T-cell lymphomas of all kinds. So, deoxycofurmycin, fludarabine, clofarabine, on and on. I wonder if it's because many of these tumors are MTAP deficient, and therefore the salvage pathways are very active in these patients.
Yeah, and one of the questions that we hoped to answer, but we couldn't get the tissue, is the pralatrexate experience in T-cell lymphoma. Are those patients that responded well, are they MTAP deficient patients? That would be a nice correlation to make.
Do you have an assay, a selective selection assay for these patients?
Yeah, there's a nice immunologic assay. We do, we have a peroxidase assay.
So our 5FU is back in favor again in pancreatic cancer. Have we looked at some of these older drugs in MTA-deficient tumor types to see if there's a differential response?
Colon cancer, unfortunately, doesn't have a high percentage of MTAP deficiency. Pancreatic cancer does, and that would be interesting to relook at in terms of using that combination. Yeah.
So the computational biology community has been very excited about MTAP deficiency because they can predict metabolic networks, and this is the classic example that they always use to say, can we predict synthetic lethality? And I have to say, personally, I've been under-impressed with what they've come up with. So I thought I would ask you, as someone who's thought a lot about this, do you have favorite, your beautiful work has really been around existing drugs. If you could start from scratch, is there another enzyme that one might be able to take advantage of in MTAP deficient cells? I'm just wondering if you have any thoughts on that.
No. It's certainly there are some inhibitors of polyamine biosynthesis, and maybe there are some possibilities there. We haven't really thought much about it yet. Yeah.
I think the question that was just asked preempts mine, but the old vessel gag of ancient times was an inhibitor of polyamine metabolism. And the question then is, would that drug or other polyamine inhibitors that were developed later, would they increase the toxicity of thioguanine because of reduced MTA?
Yeah, we thought about using, the polyamine inhibitors, but really haven't done much. I think that's an interesting area to pursue. Yeah.
Joe, a lot of this depends on whether there's a real differential between normal tissue and tumors that are in the MTAP deficient tumors. In animals, how much can you escalate the dose of 6-thioguanine or 6-MP, for example, and still rescue with MTA? And would that give you a bigger advantage by doing that? I mean, really significantly escalate.
We haven't really done that experiment yet. I think that's a worthwhile experiment doing. It's clear that we can escalate it twofold at least, because the animals will tolerate half of that dose of 75 milligrams per kilogram. That's the MTD, maybe 10-20%. So that by giving twice that dose, we can rescue them completely with, So can we tolerate 3 times or 4 times the dose? Is that going to make a difference in terms of anti-tumor effectiveness? And that's what we have to see. Yeah.
In which type of breast cancer did you see the complete responses?
The incidence of MTAP deficiency in breast cancer is about 20%. The MCF7 cell is a cell line that lacks MTAP. We haven't done much in terms of breast cancer, though. It would be a nice model to look at with fluorouracil as well, but we haven't done that.
In the slide you showed where you summarize the results of the clinical result, if I read the slide correctly, you show complete responses only in breast cancer. There were 2 complete responses. And so I was wondering what type of breast cancer were these?
Oh, these are very old data, 15, 20 years ago. So I don't think we knew what the subgroups of breast cancer were in the 15 years ago. Don't know.